When we look at living organisms, from bacteria to plants to humans, it's easy to think that life is driven by chemistry alone. But at its core, life is driven by information. Every cell in your body is constantly reading instructions, interpreting symbols, and making decisions based on molecular messages. This video is about one of the most elegant systems biology has ever produced, the genetic code, and specifically how tRNA acts as a translator that turns genetic information into living structure. What you're seeing on this slide, a DNA strand transitioning into RNA, represents a fundamental idea: information flows. DNA stores it, RNA carries it, and proteins bring it to life. Today we'll break down how that translation happens, why it's incredibly precise, and why even tiny errors can change health, identity, and evolution itself. Here we zoom in on messenger RNA, or mRNA. Unlike DNA, which stays protected inside the nucleus, mRNA is meant to be read, and it's read in a very specific way. Notice how the RNA strand is divided into groups of 3 bases: AUG, UUU, and GGC. These groups are called codons. A codon is a 3-letter word written in the RNA alphabet, and this is critical. mRNA is never read one letter at a time. It's always read in triplets. There are 4 RNA bases: A, U, G, and C. When you group them in 3s, you get 64 possible codons. Those 64 codons encode instructions for 20 amino acids, plus signals that tell the cell when to start and stop building a protein. Also notice the arrow labeled 5 to 3. This direction also matters. If the ribosome reads the message in the wrong frame, even by 1 base, the entire meaning of the sequence changes. This is known as frame shift, and it can completely destroy protein function. So at this stage, we have language, but we still need a translator. This slide introduces one of the most underrated molecules in biology, transfer RNA or tRNA. Structurally, tRNA folds into a distinctive clover leaf shape, and that shape is not aesthetic, it's functional. At the top of the tRNA molecule, an amino acid is attached. This is the building block of proteins. At the bottom highlighted here is a 3-base sequence called the anticodon. Here's the key idea. The anticodon on tRNA is complementary to a codon on mRNA. That means tRNA acts as a decoder. It doesn't just carry amino acids randomly, it ensures that the correct amino acid is delivered for each codon in the message. Each type of tRNA is highly specific. One tRNA carries glycine, another carries leucine, another carries phenylalanine. This specificity is enforced by enzymes called amino acyl tRNA synthetases, which are themselves incredibly accurate. Without tRNA, the genetic code will remain abstract. tRNA is what makes information physical. Now we see the moment of translation itself. Here, an mRNA codon aligns with a complementary tRNA anticodon. Notice the hydrogen bonds forming between the 2 bases. These are weak individually, but collectively they create strong specificity. Base pairing rule apply here. A pairs with U, G pairs with C. If the bases don't match, the tRNA simply won't stay bound long enough for translation to proceed. This is one of biology's major error prevention mechanisms. Incorrect tRNAs are rejected. Only properly matched codon-anticodon pairs are stabilized inside the ribosome. This fidelity matters enormously. A single wrong amino acid can change how a protein folds, and protein folding determines everything from enzyme activity to cell signaling to brain development. So at this scale, far smaller than anything we can see with the naked eye, accuracy is life-defining. Now we zoom out to the ribosome, often described as the cell's protein factory, but it's more accurate to call it a molecular assembly line. Inside the ribosome, multiple tRNAs dock sequentially. Each one brings its amino acid guided by codon-anticodon pairing. As each amino acid arrives, the ribosome catalyzes the formation of a peptide bond, linking amino acids together into a growing polypeptide chain. This chain doesn't just grow randomly. Its sequence, the precise order of amino acids, determines how the protein will fold. And folding determines function. For example, hemoglobin folds to carry oxygen. Enzymes fold to catalyze reactions. Neurotransmitters, receptors fold to detect signals. So when we say genes code for traits, what we really mean is genes code for protein sequences, and protein sequences create biological behavior. This slide highlights something essential. Translation is not random. Every protein begins with a start codon, almost always AUG, which also codes for the amino acid methionine. AUG tells the ribosome, begin here. From that point on, the ribosome reads codons one by one until it reaches a stop codon. Stop codons, UAA, UAG, and UGA, do not code for amino acids. Instead, they signal termination. When the ribosome encounters one, translation ends and the protein is released. This system defines protein length, which is critical. A protein that's too short may be non-functional, too long, and it may fold incorrectly or interfere with other cellular processes. This slide reinforces a major theme of biology. Life depends on regulated boundaries, not chaos. Here we zoom out, from ribosome to cell to organism. What's remarkable is that the same genetic code is used by almost all life on Earth. A codon that codes for leucine in bacteria codes for leucine in humans. This universality tells us something profound. All life shares a common molecular language. Small changes, mutations, can alter that language. A single base change can substitute one amino acid, shorten a protein, eliminate function entirely. Sometimes these errors cause disease like cystic fibrosis or sickle cell anemia. Other times they create variation that drives evolution. So the genetic code is not just about biology, it's about identity and change. Every emotion you feel, every thought you have, every moment you make depends on proteins that were once just lines of code, decoded faithfully by tRNA. So, thank you for watching and see you next time.